Good morning, everyone. I'm delighted to welcome you all today. My name is Dr. S.K. Singh, and I hold a PhD in this fascinating field. Today, we'll be exploring the principles of liquid chromatography, mass spectrometry, or LCMS for short. Over the course of this presentation, we'll cover the fundamental concepts, how this technique works, and its applications in various scientific areas. So, let's get started and dive into the world of LCMS. Let's take a closer look at what LC-MS actually is. It stands for liquid chromatography, mass spectrometry, which is a powerful hyphenated analytical technique. Essentially, it combines two different methods to give us a more complete picture of our samples. First, we have liquid chromatography, or LC, which is responsible for separating the mixture into its individual components. It helps us isolate what we're interested in from complex samples. Then we have mass spectrometry, or MS, which identifies and quantifies these components based on their mass-to-charge ratio. This dual approach makes LC-MS incredibly versatile and precise. You'll find LC-MS widely used across various fields. In pharmaceutical analysis, it helps in drug development and quality control. In bioanalysis, it's essential for studying biological samples like blood or tissues. It's also heavily relied upon in environmental and clinical studies, where detecting trace levels of chemicals or biomarkers is critical. Overall, LC-MS is a cornerstone technique because it combines separation and identification in one integrated process, enabling us to tackle complex analytical challenges efficiently. Separation principles in chromatography are primarily based on different properties of the molecules we're analyzing. One key factor is polarity, how polar or nonpolar a compound is, which influences how it interacts with the stationary phase. Another important aspect is the partition coefficient, which describes how a compound distributes itself between the stationary phase and the mobile phase. Additionally, the way molecules interact with the stationary phase plays a big role in their separation. A common mode we often use is reverse phase chromatography, which typically involves a C18 column. In this setup, nonpolar compounds tend to be retained longer, while more polar molecules pass through more quickly. Once separation occurs, we generate a chromatogram, which visually displays the different compounds based on their retention times. Moving on to the principles of LC-MS, or liquid chromatography mass spectrometry, it's built on two main processes. First, we have the chromatographic separation, where compounds are separated in the liquid phase. Then, we detect them based on their mass-to-charge ratio, or MZ. The workflow starts with a sample that goes through the LC system, where separation happens. Next, the separated molecules are ionized, making them detectable by the mass spectrometer. The ions are then analyzed in the mass analyzer, which sorts them based on their MZ values. Finally, the detector records the data. From this process, we get two key outputs: the retention time, which tells us when a compound elutes from the column, and the mass spectrum, which provides detailed information about the molecular weight and structure of the analyte. These combined data points are essential for identifying and quantifying compounds in complex mixtures. Let's talk about ionization techniques, starting with electrospray ionization, or ESI. ESI is a soft ionization method, which means it gently transfers molecules into the gas phase without breaking them apart. This technique is particularly suitable for polar compounds, making it very useful in analyzing molecules like peptides and proteins. Now, moving on to APCI, or atmospheric pressure chemical ionization. This method is better suited for less polar compounds. One of its key advantages is that it produces molecular ions with minimal fragmentation, so you get a clear picture of the molecular weight without a lot of breakdown products cluttering the spectrum. As we continue, remember that these principles are fundamental to understanding how different ionization techniques are chosen based on the chemical nature of the analyte. They help us optimize detection sensitivity and accuracy in mass spectrometry analysis. Let's take a closer look at the interface, which is a crucial part of LC-MS coupling. Its main role is to convert the liquid sample coming from the chromatography system into gas phase ions that the mass spectrometer can analyze. This process involves removing the solvent, which is essential because excess solvent can interfere with the detection and reduce sensitivity. At the same time, the interface helps maintain atmospheric pressure ionization, allowing the ions to be efficiently transferred into the mass spectrometer. This step is critical for achieving high sensitivity and accurate results. Moving on to the mass spectrometer components, we have several key parts. The ion source, which can be ESI or APCI, generates ions from the sample. These ions are then guided through ion optics, which focus and direct them toward the mass analyzer. The mass analyzer separates the ions based on their mass-to-charge ratio, and the detector records the ions that reach it. Finally, the data system processes this information to create the spectra we analyze. The main components of the overall system include the LC system, the interface we just discussed, the mass spectrometer itself, and the data system that handles all the information. Now, focusing on the LC system components, we have a high-pressure pump that delivers the solvent steadily and precisely. The autosampler injects the sample into the system, ensuring consistency and reproducibility. The column, which can be C18, C8, or other types, is where the separation occurs based on chemical properties. And the mobile phase, typically a mixture of water and organic solvents, helps carry the sample through the column. This overview ties everything together, emphasizing how each part plays a vital role in achieving accurate, sensitive, and reliable analytical results in LC-MS. Let's walk through the working mechanism of our mass spectrometry system. First, we start with sample injection, where the sample is introduced into the system. Next, the sample undergoes liquid chromatography separation, which helps us isolate compounds based on their interactions with the stationary phase. After separation, the compounds are ionized, turning them into charged particles suitable for mass analysis. Once ionized, these ions are transferred through the instrument to the mass analyzer. The mass analyzer then separates ions based on their mass-to-charge ratio, or MZ, which is crucial for identifying and quantifying compounds. Following separation, the ions are detected, and this results in signals that are processed into data. Now, when we look at advanced systems, we see options like LC-MS-MS, which is tandem mass spectrometry, providing high specificity and sensitivity. High-resolution MS, or HRMS, offers very precise measurements, allowing us to distinguish compounds with very similar masses. UPLC-MS combines ultra-performance liquid chromatography with mass spectrometry for faster, more efficient separations. Talking about mass analyzers, we have several types: quadrupoles, ion traps, TOF, or time-of-flight analyzers, and orbitraps. Each of these separates ions based on their MZ ratio, but they do so with different principles, offering various advantages depending on the application. Moving on to the detector and output, we use devices like electron multipliers, or MCPs, which amplify the signals for detection. These detectors produce a total ion chromatogram, or TIC, which shows the overall ion signal over time, and a mass spectrum, giving us detailed information about the ions present. This setup allows for high sensitivity detection, ensuring we can detect even trace levels of compounds. Finally, this overview ties into our overall instrumentation, highlighting the key components and how they work together seamlessly to give us accurate, reliable data in our analyses. Let's take a moment to focus on why instrument qualification and data integrity are so crucial. They're required not only for regulatory compliance, but also to ensure the reliability of our data. When we talk about instrument qualification, or IQ, we're referring to a series of steps that verify our instruments meet specific standards. This includes design qualification, or DQ, installation qualification, IQ itself, operational qualification, or OQ, and performance qualification, or PQ. These steps are essential to confirm that our equipment is properly installed, functioning correctly, and consistently delivering accurate results. Now, these requirements are not just best practices, They're mandated under regulations like FDA 21 CFR and USP 1058. Following these guidelines helps us stay compliant and maintain the integrity of our data. Ensuring data integrity is fundamental because it underpins the trustworthiness of our results. To achieve this, we focus on validation parameters such as specificity, linearity, accuracy, precision, and limits of detection and quantification, LOD and LOQ. Additionally, robustness testing confirms that our methods remain reliable under varying conditions. All these measures work together to ensure our instruments are reliable and our data is trustworthy, which ultimately supports our overall goal of delivering high-quality, compliant products. Now, let's take a look at some of the key applications and examples where our techniques make a real difference. In the pharmaceutical industry, one common application is drug assay, such as measuring paracetamol levels. This helps ensure proper dosage and safety for patients. Moving on to bioanalysis, a typical example is monitoring plasma drug levels using multiple reaction monitoring, or MRM mode, which provides highly sensitive and specific analysis. In the environmental sector, our methods are crucial for detecting pesticides at trace levels, helping us monitor and protect ecosystems from harmful chemicals. Lastly, in clinical settings, vitamin D analysis is a great example. Accurate measurement here is important for diagnosing deficiencies and guiding patient treatment. Overall, these examples highlight the versatility of our approach across different fields, from pharmaceuticals to environmental monitoring and clinical diagnostics. It's all about providing precise, reliable data to support critical decisions in health and safety. Let's take a closer look at the advantages and limitations of this analytical technique. Starting with the advantages, one of the key strengths is its high sensitivity, often reaching parts per billion levels. This means we can detect very small amounts of substances, which is crucial in many applications. It also offers high specificity, allowing us to distinguish between compounds even if they are very similar. This makes it particularly useful when analyzing complex samples. Another great point is that it's suitable for non-volatile compounds. Expanding the range of substances, we can analyze beyond just volatile ones. And finally, it enables both qualitative and quantitative analysis simultaneously, saving time and providing comprehensive information in a single run. Now, turning to the limitations, one major factor to consider is that the instrumentation can be quite expensive. This means significant investment is needed to set up and maintain the equipment. We also face challenges with matrix effects, such as ion suppression, which can interfere with accurate measurements depending on the sample matrix. Additionally, operating this technique requires skilled analysts who understand the nuances of the process, adding to the resource requirements. So, while this method offers many benefits, especially in sensitivity and specificity, we must also be mindful of these limitations when deciding if it's the right tool for our analysis. So to wrap things up, let's talk about the conclusion. LC-MS, or liquid chromatography mass spectrometry, is truly a powerful analytical tool. It combines two essential processes: separation, which is the liquid chromatography part, and identification, which is handled by the mass spectrometry. This combination allows us to analyze complex samples with both precision and confidence. You'll find LC-MS plays a critical role in fields like pharmaceuticals and clinical research. Its ability to accurately identify and quantify compounds makes it indispensable. But of course, working in these areas requires strict regulatory compliance to ensure safety and efficacy. Overall, LC-MS's strength lies in its ability to deliver detailed, reliable data, making it an essential part of modern analytical workflows. For further support, please feel free to contact us. We're here to help with any questions or additional information you might need. And with that, I want to thank you all sincerely for your time and attention today. Your engagement is truly appreciated, and I hope you found this presentation valuable. Remember, we're just a message or a call away if you need anything moving forward. Thanks again, and have a great day.